Hi, my name is Austin Kruger, and this summer I've had the great pleasure of working with Dr. Terry Hines here at the University of Toledo. Over the summer, we researched glucocorticoids and prostate cancer, and specifically we looked at glucocorticoids as it relates to prostate cancer in black men. For some background on our study, prostate cancer is one of the most common cancers found in men in general. However, it is more common among black men than it is among Caucasian or Asian men, and tends to have a worse prognosis in this population than in the others. Now, while a part of this is probably due to societal factors, less access to healthcare, less access to the means of a healthy lifestyle, this does not seem to account for the entirety of the difference between these different populations. So our lab has been looking at different signaling pathways within prostate cancer cells that could be different between black men as opposed to Caucasian and Asian men. Specifically, we're looking at the intersection between receptor tyrosine kinase pathways and glucocorticoid receptor pathways. Now, the reason why we look at these specifically is because these receptors seem to play a role in a transition of prostate cancer towards a more aggressive form. Specifically, within early stages of the disease, prostate cancer is very sensitive to levels of androgens within the system, and the prostate in general is, but prostate cancer specifically in its earliest stages needs androgens in order to grow. So our first line of treatment against a disseminated prostate cancer is androgen ablation through either chemical or physical castration. However, this inhibition on the growth of prostate cancer is almost always temporary. Eventually, the prostate cancer develops a resistance to androgen ablation and develops a more progressive form that we call castration-resistant prostate cancer. Now, it's thought that receptor tyrosine kinases play a significant role in this transition, hence our interest. Receptor tyrosine kinases are receptors found within the plasma membrane of cells that, when they bind to their agonist, dimerize and initiate signaling complexes that do a wide variety of things within the cell. And we'll get down into the specific ones that actually relate to prostate cancer in a second. The glucocorticoid receptor, on the other hand, is a nuclear receptor that binds to its ligand and then translocates into the nucleus to initiate most of its signaling, which is a bit of a simplified form. Actually, the glucocorticoid receptor exists as two isoforms, GR-alpha and GR-beta. GR-alpha does bind ligands and acts in what might be considered a more canonical pathway. However, GR-beta actually does not bind most of the ligands that we associate with the glucocorticoid class, but instead seems to have constitutive activity that's based more on its expression level within the cell and possibly interaction with other signaling pathways as well. Within prostate cancer, we're interested in the glucocorticoid receptor because GR-alpha seems to have some protective effects. It promotes apoptosis and inhibits the growth and proliferation of cells, and particularly it does this through inhibiting receptor tyrosine kinase pathways. On the other hand, the GR-beta isoform tends to promote the progression of prostate cancer cells. And it does this largely by acting as a negative inhibitor, a dominant negative inhibitor of GR-alpha. So the greater the expression of GR-beta you have within the cell, the less activity you get from GR-alpha and the less inhibition of receptor tyrosine kinase pathways that we see. Now, we're going to go even more specific than that, but I'd like to do that with an image. The receptor tyrosine kinase pathway that we looked at in our lab is the PI3K AKT pathway. In this pathway, an activated receptor tyrosine kinase complex binds and activates PI3 kinase, which converts inositol diphosphate to inositol triphosphate within the plasma membrane. Inositol triphosphate acts as a docking site for AKT and a variety of other proteins as well, but it acts as a docking site for AKT in particular and activates this protein. AKT mediates many effects within the cell, but most importantly for our consideration is that it promotes growth and proliferation, survival, metabolism, and proliferation. And it's thought that this is one of the primary pathways that allows for escape from inhibition of the androgen pathways, which are normally the ones that sustain cell growth and proliferation within the prostate. Glucocorticoids come into this signal as in a variety of different ways, but primarily through P10. GR-alpha increases expression of P10, which is phosphate-intensive homolog deleted on 10, which is a phosphatase that converts IP3 back to IP2, and therefore inhibits the activity of the PI3K AKT pathway. Now, there are other actions And there is some thought that glucocorticoids and RTKs have a crosstalk in their expression profiles, which is one of the things that our data specifically looks at. But also, GR beta increases expression of certain actors within this and certain isoforms of, for instance, AKT that are more prone to promoting malignancy. So going on down to the experiments in our data. One of the first things that we did is we took our African American cell lines and treated them with different receptor tyrosine kinase agonists to see how this would affect the expression of GR alpha and GR beta within the cell. In these images, the first image that you see in each class is, for context, and hopefully you're not colorblind today, the green cells are imaging for GR beta. We are doing immunohistochemistry, immunostaining using fluorescent markers. We permeabilize the cells, put in an antibody for GR beta, then put in a secondary antibody with a fluorescent marker so that we can image it. So the first line and what you see in green is GR beta expression. Red is GR alpha expression. The third image is a merge of the two, which, because GR alpha expression is not showing up as brightly in these images, mostly looks green. And the final one is a merge of these images plus DAPI, which is a stain for the nuclei, and a bright field microscope image. Now, this would be a good time to talk a little bit about the cells that we're working with. E006AA PAR is a cell line that's still androgen sensitive. So this represents more the early phase of prostate cancer. E006AAHT is a cell line derived from the PAR cell line, done by depriving the cell line of growth factors and then seeing selecting the cells that can survive in that environment. So it represents more the advanced stages of prostate cancer. And as you can see, we have control, insulin, PDGF, and EGF. And what we do with these images is we put them into a software called ImageJ that can measure the amount of pixels within a particular area and give us a density, which acts as a good proxy for the expression level within a cell. And that's where our next data comes from. This is the expression of GR beta to GR alpha normalized against the control. And as you can see, insulin really doesn't do anything in terms of this ratio within the cell. However, both PDGF and EGF seem to increase the amount of GR beta compared to GR alpha. While both of them seem to have this effect, only PDGF, though, reaches a statistically significant effect level within the cell. Now, this is actually very good data and very consistent with other findings that my lab has found. Prior to my arriving there, they looked at the different levels of receptor tyrosine kinases within cells. And one of the things that they found is that the insulin receptor is expressed at higher levels in Caucasian men, but is expressed at very low levels in black men, while PDGF and EGF are both expressed at much higher levels in black men than they are in Caucasian or white men. Caucasian or Asian men. So we're seeing a continuation with previous data that's really nice to see. So that's the PAR cells. If we look down at the HTs, we see a very similar pattern. Again, everything is normalized against control. Insulin doesn't seem to have very much of an effect towards shifting things, in terms of shifting things towards GR beta, but both PDGF and EGF tend to push cells towards a higher level of GR beta expression. Now, what's a little bit misleading in this, it looks like there's actually a bigger effect size with PDGF in this one than there was within PARs, but it doesn't reach statistical significance on any of these factors within HT. And that's likely due to a number of factors. One, we had a fairly small sample size, and two, the standard deviation for PDGF in particular was much higher with the HTs than it was for the PARs, which again is probably due to a small sample size and possibly having one or two outliers within our data set there. However, my suspicion would be that if we continue these experiments, you'll see that both PDGF and EGF will reach statistical significance, which is something that my lab is planning on doing. I don't know whether I'll be there or not, only another week and a half to go, but it'll be cool to see once they actually get to the data. So that's pretty much it for what we're looking at here. And actually, I'm going to go back for a second. So part of the story that this really functions well in is our operating hypothesis is that GR beta is expressed at much higher basal levels within African American men than it is within Caucasian men. And that accounts in part for the increased prevalence towards prostate cancer and the increased severity of the disease. The fact that PDGF and EGF are expressed at higher levels, that PDGF and EGF receptors are expressed at higher levels within black men, and that both of these receptors seem to increase the level of GR beta within the cell, fits that story very well and suggests a very good avenue that we can look at for targeting treatments towards African American men specifically that look at the PDGF and EGF signaling pathways. Okay, so that's pretty much it for that particular assay. I also had a small side project in which we were looking to see whether these cell lines are migratory. The two African American cell lines that we're working with are relatively new. There's not very many labs working with them. They were only derived a couple of years ago, and that's really cool for our project, but it also means that there's some gaps in our knowledge. And my PI wanted to know if these are migratory because that provides the basis for doing functional studies. So we could treat cells with inhibitors and see if it slows them down in terms of how quickly they can move, which is a proxy for malignancy and for metastasis in some cases. However, we need to be able to differentiate between cells migrating and cells simply proliferating. And so that's what this study looks at. And as you can see, there's a fairly significant closure of the gaps here. The way that this study works is I take a 1,000 microliter pipette, run it along a confluence culture of cells, and then take images over the next couple hours. The important thing, though, is the time frame that these images are being taken during. The dividing time for these cell lines is about 21 hours. It's a bit more for the HT cells and the PARs, but in that general category. The fact that we see significant closure of the wounds made in the cultures at 11 hours tells us that these cells are in fact migrating and that the closure is not simply due to increased proliferation of cells. So my lab will be able to take this information and continue to run with it after I've left and continued back with medical school. So conclusions from our study. Expression of GR beta appears to be promoted by EGF and platelet-derived growth factor. However, only PDGF produces a statistically significant increase, and this statistically significant increase was only seen in the PAR cells. This is consistent with previous findings that EGFR and PDGFR are more highly expressed in cells from black men than those from Caucasian men. Additionally, both the HT and PAR cell lines are migratory. Thank you very much for listening to this presentation. I hope that you've learned something from it.